Yeah, good afternoon, everyone. My name is Pedro. I'm a postdoc at SAIAB, and it's a great pleasure to chair this section with the guest speakers. And now I will briefly introduce us to the next speaker, that is Elisabeth Henschel, a friend and colleague from Brazil, from the University of Rio, Federal University of Rio de Janeiro. She's finishing her PhD next month, and this was one of the reasons we put her name to be voted to be selected for this talk, because she had a lot of similar things shared with the South African and SAIAB students, many of them finishing now their PhDs. And one of the reasons is what Elisabeth, I know her a lot. So what she have achieved in the past 4 years during her PhD as a woman, a researcher on taxonomy of fishes in Latin America. And there are a lot of similarities and challenges in doing that kind of research in Latin America, in being a woman, and in South Africa. So I think it will be a great opportunity to listen to her, and that's it. And there's just to make it clear, she's a specialist on parasite catfishes in the Neotropical region, and it would be lovely to listen about the importance of taxonomy and systematics in a changing world, in Africa, Latin America, everywhere.
Well, hello everyone. Good morning for those who are in Brazil or in the US, I don't know, and good afternoon to you all that are in Africa. So I am Elisabeth Henschel. As Pedro said, I work with neotropical fishes, but I was invited here to talk to you, to talk to other SAIAB grad students about the challenges we face. So I'm gonna share my screen right now. And I would like to be sure that, oh God. Are you all seeing my screen, my full screen? Pedro, please tell me if it's okay.
It's okay.
Okay, so can I begin?
Yes.
So today I would like to have a conversation with SAIAB's graduate students about challenges we face as graduate students in emerging economies, and all the challenges we face and all the opportunities I had. I consider myself a very lucky person, so I would like that all other grad students from wherever they are had the same opportunities I had, and that's why I would like to share a little bit more of my journey with you all. So as Pedro said, I am a PhD student in genetics at the Federal University of Rio de Janeiro in Brazil, and that's it. I'm very excited and very honored to be here talking to you all on your student symposium. So those are the topics we're going to cover today. So I will begin talking a little bit more about the importance of taxonomy and systematics to society. And then I'll go for a little bit of the similarities between Brazil and South Africa, and where we are as emerging economies and where we early career scientists are studying the biodiversity. And then we'll share a little bit about my short career, and then the final intervention will question our role as graduate students in the future of taxonomy and systematics. So let's begin. I'm sorry if I'm talking a little bit fast because I'm anxious and nervous, but I hope you all can understand what I'm saying. So what is a biodiversity crisis? Biodiversity crisis is the rapid loss of species and the rapid degradation of ecosystems that affect all of our natural resources. So we are now entering the 6th mass extinction, and we are losing diversity as a whole, diversity within species, between species, and within ecosystems. So we are losing variability that is the base, the foundation for all life. So probably until 2050, half of our species may face extinction. So this is our scenario right now, and probably the biggest crisis we are facing as humans. And now we are also seeing the biggest decay of life that ever happened. So life is declining faster than any other time in human history. especially after the Industrial Revolution, when human activities greatly increased. So here on this graph, we can see how the biodiversity has been declining since 1970, I mean, the last 40 years, and population size of vertebrates dropped 68%. So this is a very degrading scenario in terms of biodiversity. So here we can see by this estimate that we still have around 8 million species of animals to be discovered. So science still doesn't know around 8 million species of animals, and we don't know what these animals can tell us about the future, about our lives, about nature. So if we lose all this biodiversity, our future is also compromised. So, and when compared to forests and oceans, freshwater is the most endangered environment. So healthy rivers, lakes, and wetlands provide huge benefits to people all around the world for farming, industry, drinking water, and even for our social lives. But although fresh water only represents 3% of our land surface, 1 in 3 freshwater species are threatened with extinction. And freshwaters are home to 10% of all animals and more than 50% of all fish species. So we can think about how worrying it is for us to have fresh water at this level of danger. And in this picture, we can see a dam built in the Madeira River, which is like one of the biggest rivers of the Amazon River basin. And I selected this picture because I personally relate to the history of this dam, because although I live in Rio, which is in southeastern Brazil, my father lives in the city where this dam was built. So I remember when I was a teenager, around 12, 13 years old, I used to go there to the city where he lives, and life around the river was extremely different. So the landscape was much more interesting, much more full of life, not only of animals, but also the social life, the community around the river was much more rich. And after this dam was built, not only the landscape changed, but also the economy. I mean, the city changed a lot with the construction of the dam. So this is something for us to think about. Our impacts in nature affect not only nature itself, but also our lives. So what is our role as taxonomists and systematists in this degrading scenario we are living right now? So we are seeking answers and solutions to our real world problems. And taxonomy and systematics are the foundation for all other fields of biology and for its applications. So these fields of knowledge, taxonomy and systematics, allow us to access biodiversity, to understand the base of adaptation, to access inter and intrapopulational variation, and to understand the complexity of evolution as a whole, considering time, space, and shape. And the knowledge we can generate as taxonomists and systematists can be used for conservation, to provide data on economically or culturally relevant species, can be applied to agriculture or public health. And when we think about public health, I cannot not mention the current pandemic we're living and all the understanding that systematics provided to us. So nowadays we know that this new COVID virus was not manufactured in a lab. It probably arose from a wildlife species. And this was an understanding provided by systematics. And systematics and taxonomy also provide insights on how to deal with invasive species, to understand speciation mechanisms and phenotypic plasticity. And where are we living in an emerging economy, being early career scientists and conducting research on biodiversity? So now I would like to highlight some similarities between Brazil and South Africa. So this is the Living Planet Index by region, and this is a report published every 2 years by WWF and in collaboration with the Zoological Society of London. And it measured the states of the world's biological diversity based on population trends of vertebrate species from all over the world. So here we can see that Latin America, the Caribbean, and Africa share the same horrifying status. So we're basically the two continents that are facing the worst impacts of biodiversity loss. So this represents the fall in species abundance in the last 50 years. So when compared to all other continents, Latin America and the Caribbean and Africa are suffering the worst impact. And this is the graph I developed on the World Bank database, and I decided to compare the research and development expenditure based on percentage of the gross domestic product between Brazil and South Africa and the major world powers. So I thought of 3 countries that I know that are investing lots of money in science and research. So I decided to compare with Germany, the United States, the US, and China. So here we can see that Brazil and South Africa are very, how can I say that? We are investing much less percentage of our domestic products than the countries that are considered global potencies in science. And actually here in Brazil, we can see that our funding for science is declining over the last years. So this is a situation that all my colleagues and friends and family that are here watching this lecture will relate to. And the 2 biggest challenges to taxonomy, according to Britain Collaborators 2020, are the inadequate funding and the lack of succession planning, training, and recruitment into permanent positions of competent taxonomists. And these effects are accentuated in emerging economies where funding is even more scarce. So if there is a global tendency to cut funds from taxonomy and systematics, be sure that this situation is even more accentuated in the countries where we live, in emerging economies. So first, inadequate funding affects all stages of research, from lab maintenance, for example, buying reagents, being able to clean our workspace, to availability of scholarships, funding for early career scientists, and our ability to conduct field work. Inadequate funding leads to the second consequence that affects especially early career researchers and graduate students that will face a constrained job market once we finish our PhDs. So this is a very worrying situation for us. But here, I would like to highlight some positive similarities between Brazil and South Africa. So there is usually a tendency to consider Latin America and the Caribbean and Africa as completely uniform continents. So people usually think, oh, Latin America does not invest in science. Africa does not invest in science. But we have inner differences with our neighbors. So here on this graph, on the y-axis, we can see the amount of researchers per million inhabitants, and on the x-axis, the research and development expenditure as percentage of the gross domestic product. And the size of the ball refers to the absolute amount of money dedicated to research and development. So here we can see that Brazil is probably the country that invests the most percentage of our gross domestic product on science and research. Although Argentina seems to have more researchers per million inhabitants, we are investing more. And the same happens with South Africa. So when compared to its neighbors in the sub-Saharan Africa, South Africa is investing more. So it means that us researchers in Brazil and South Africa are having more opportunities than our neighbors. And this is something for us to think about. So now I would like to share with you a little bit of my journey and the challenges we faced and the opportunities I had, because when this talk ends, I would like to hear from you what you think about it, what you would like to do, how you feel about your own PhDs or masters. So all my academic education took place at the Federal University of Rio de Janeiro in the same lab. So it's like a 10-year relationship that I'm very proud of. So I did my bachelor's degree in biology with an emphasis in zoology. And then I got my master's degree in biodiversity and evolutionary biology. And now, as Pedro said, I'm finishing my PhD in genetics. And I had the opportunity to spend a couple months at the American Museum of Natural History. And in 2018, I got a National Geographic Early Career Grant, so I can be considered an explorer. And now I'm currently enrolled on their Explorer Educator Exchange, which is basically a co-training between an explorer and a certified educator. So together we think about and train each other on educational possibilities, projects, and we think about education. So the first part of my academic education was from 2011 to 2015, when I joined. I was 17 years old when I joined the Laboratory of Systematics and Evolution of teleost fishes under Dr. Wilson Costa's advising, and he's my advisor until today, so it's a long-lasting relationship. And shortly after I joined the lab, I did my first expedition in the Amazon with Dr. Pedro Bragança, who just introduced me. And this was when I fell in love with Amazonian fishes. So this is why I think it's so important for undergrad students to do field work and to be on the field, because that's when you see the fishes or whatever you are working with, and where you can collect additional data that are not available in biological collections, for example. And the field work is where you have the opportunity to make friends for life, establish collaborations for life. And I don't know, I'm very passionate about field work. And basically for me is where everything happens. So I can see that someone said something on the chat, and I would like to ask Pedro to interrupt me if it's something important. But so that's it. That's where you can see other animals, where you can be in touch with the organism you're studying, and sometimes you can face death risk, but it's okay, everything is fine. And so during my undergrad students, I conducted 5 expeditions to distinct locations in the Amazon and Pantanal, which Pantanal is the biggest wetland we have here in South America. And Wilson provided me an intensive training. Wilson's my advisor, provided me an intensive training on morphology. So basically I was focused on osteology, meristics, and external anatomy, and applied this knowledge to taxonomy. So during these 5 years, I collected, Pedro and I and other colleagues collected several populations of these miniaturized catfish from the Amazon. And although they all look apparently similar, so they share the same external anatomy with its basic coloration pattern, small size, and due to this, people tend to identify them all over the Amazon River basin as the same species, but it happens that they are not. So with this dense training on morphology, I could identify a new species of miniaturized catfish. That was my final work as an undergrad student, and it turns out to be this species description that was published in 2016. And then the second part of my academic education took place from 2015 to 2017. That was my master's. So I kept doing field work in the Amazon and Pantanal. I mean, seeing those animals is just amazing. It's one of the best parts of the whole experience. And I pursued my master's on biodiversity and evolutionary biology. And after I had this intensive training on morphology, this was when I first had the chance to be educated on molecular systematics. So all of those populations of miniaturized catfish that I talked to you about, they happen to be a new genus. So we used a DNA-based phylogeny to position this new genus of miniaturized catfish within the family. So molecular systematics was very important in this case because those fishes share a lot of reductions and a lot of morphological convergence. So a morphological phylogeny usually does not provide us a good phylogenetic hypothesis. But of course, morphology was definitely not forgotten, and it is a very crucial part of this work. So it was fundamental for us to establish the new genus diagnosis. So here I just put the osteological illustrations of this work where we described this new genus of Amazonian miniaturized catfish. And in 2017, I started my PhD in genetics. And now I am further applying my knowledge on taxonomy and systematics into the morphofunctionality and ecology of kangaroos. And kangaroos are a very special group of catfishes. They are mostly located in the Amazon, but there are other kangaroos outside the Amazon River basin. And they are known for being parasitic. So kangaroos can feed on blood, mucus, scales, parts, and even on organic decomposing matter of other fishes. So they are parasites. And they are very known here for sometimes accidentally entering the human urethra. So there is a lot of fantasies and history surrounding kangaroos. It's a very interesting group of catfish. But what I would like to say here is that the PhD are probably the 4 most decisive years of our lives. So you gotta trust yourself, your project, and your advisor. So if your advisor agreed to advise you and build a project together with you, I'm sure that they trust you. So this is something for us to always keep in mind, even when things appear to go wrong. And in 2018, I was very lucky and I made a huge effort into getting this National Geographic Early Career Grants. So what I'd like to say here is that since sometimes we don't have sufficient funding for all aspects of our research, we got to think of complementary funding, look for other sources of funding, not only NetGeo, of course, there are many others, local and global alternatives to fund distinct aspects of our research. So the more you apply, the more chance you'll have to be granted. So, and do not be discouraged by an eventual rejection, because I submitted other projects that were also rejected. And your success depends not only of the quality of your research proposal, but also of what other applicants are proposing and the amount of available grants and many other aspects we cannot control. But the thing is that being funded by an NGO or wherever provides you new approaches to field work, because you need to think about what is interesting not only for your research, but to the institution that is funding you, provides you new career opportunity, networking, and the most important, I would say, is public outreach, because these institutions are usually committed to reach a larger audience. And this is an example of what happened. So on this NetGeo funded fieldwork, I described a new species of miniaturized catfish from the Amazon, not the ones I was talking about, but a distinct one. And NetGeo Brazil decided to do an article about it, which is very nice. And in 2019, I did part of my PhD at the American Museum of Natural History. And this is something that I think you should keep in mind as well. So I'm very thankful to my advisor there, Dr. Scott Schaefer. And working in a foreign institution allows you to have access to specimens you would not have access in your home institution. So for example, there I had access to kangaroos and catfishes from other countries in South America that I wouldn't have access here in Brazil. And you'll also probably experience a new infrastructure. So in my case, I had a chance to work with CT scans. So I never worked with CT scans before, and it is a more modern approach to osteology and to morphology in general. And you'll also have the chance to establish new collaborations, to visit other institutions. And I don't even need to mention the cultural enrichment that you will carry for your life. So this is something that if you have the chance, please do not miss this. And this is an example of a CT scan. And this work regards a new species I just described. Oh my God, how did I take this off? Here, this is a new species of kangaroos that my collaborators and I just described. And this kangaroo, we only had a single specimen of it. So therefore, in this case, CT scan was very, very important for us to have access to data of inner morphology. So before we go to the last part, I would like to do some important remarks. So first, being a woman in the field. So if you, my listener, is a woman and doing field work, cheers. I'm with you. You have the guts, because as women, we experience the field in a much more heavy way than our male colleagues. So during this 10 years of field work in the Amazon, I had although it was very wonderful, I had many uncomfortable experiences, from harassment to people being disrespectful to me. So, for example, once I was funding one of this NetGeo funded trip. I mean, the money was with me, and we were hiring someone to work for us. And this guy didn't even look at me in the face. So I had to be with my colleagues. So I had to communicate everything that I wanted to happen on the field to my male colleagues for them to communicate with the people we were hiring. So being a young woman in the field is definitely difficult. So it's a challenge. And second is the importance of a solid knowledge on taxonomy and systematics based on morphology and DNA. So sometimes people think, oh my God, DNA is the answer for all of our problems, and it is not. There was even a time when people thought that DNA would fasten the pace of a species description and of taxonomy, but that's definitely not the case, because DNA helps us a lot in terms of cryptic species and groups where we have lots of morphological convergences, reductions in groups where homology is very hard to access. But morphology still got a lot of new information to our studies. So not only you can approach some understudied structures such as muscles, nerves, histology, but also more the traditional approaches such as clearing and staining specimens, and more modern approaches such as the CT scans. Osteological techniques can still reveal new structures and bring us evolutionary insights. So let's not forget the importance of morphology, of good morphology to our work. And finally, I would like to reinforce the importance of us graduate students in the future of taxonomy and systematics. So here, what is our role? So I'm going back to what I said on the introduction. We will still be seeking answers and solutions to our real world problems. So in 5, 10, 20, 30 years, we will be where our advisors and supervisors are now. And we will be facing a much more devastated world. So in 2050, I hope we all have permanent positions, and I hope we all are still working with what our love. And so if by 2050, probably half of species may face extinction, I'm sure we will be experiencing an increase on social inequality and a decrease on the availability of natural resources. So we gotta be prepared, because we will be the qualified workforce to answer to this crisis, to provide basic data and information for government, NGOs, and for the public to deal with the future world that will probably, I hope not, but probably will be much worse than ours. And lastly, I would like to share some reminders for early career taxonomists and systematists. So a reminder for myself as well. So first, study the diversity of life through multiple and complementary perspectives. So that's what I just said. Let's focus not only on morphology or DNA or ecology. Let's try to approach our study in a more holistic way. And not forget the importance of fieldwork and of scientific collections. So fieldwork provides us lots of data that scientific collections do not, and vice versa. So fieldwork and scientific collections are complementary. So let's try to leave both worlds. Make good collaborations and trust your advisors. This is really important, because this is what I said as well. So your advisor trusts you. I mean, you and your advisor are like a single organism trying to succeed on this project. So, and making good collaborations also beyond your lab is also really important. And extend your limits beyond academia. This is something that I think is crucial for our future. So let's think about public outreach, because we are now living a time where public understanding of science is kind of threatened and is also endangered. So we should think of ways and work with institutions that want to translate our work to a wider audience. And I know that sometimes we think that nobody's interested in what we are doing. There is that common sense that taxonomy is boring, but it is not. Actually, the public is really interested in scientific discovery, new species. They are amazed that there are still many things to be discovered. And finally, remember that each grad student is struggling to do good work. So be kind to yours. I mean, we are in the same situation. Let's try to help each other. And oh gosh, this is on the way of my Mimi. So I know that most of us, at least I can speak for myself, blame TikTok for everything that's happening, for all the stress, heavy load of work. And we think that this fish decided to walk out of the water one day and I'll have to finish my thesis. Oh my God, what happened? I know this is common and this is part of our process. So, but I would like to finish this lecture with a positive message. So relax, we will all graduate. We might get a job, I hope so. And things take time. Every one of us has its own time, and just enjoy where you are now. I remember when I just started my PhD, I met a teacher in the hallway and she said to me, Elisabeth, please enjoy these 4 years of PhD because they will probably be the 4 happiest years of your life. And in my case, they were. I'm already getting emotional, and just enjoy because I'm sure that we will miss it, having a goal and the excitement of doing new discoveries. And I hope it keeps with us for the rest of our lives. So I would like to thank you all for being here, for listening to what I had to say. I know that I don't work with African fishes, but I hope that I could give you some insights, and I would love to hear from you. What are you facing? What are you thinking? Here is my email and my website. And I would like to especially thank Dr. Albert Chacona and Pedro for inviting me to this event. This is amazing. It's such a nice experience. And to thank all my advisors for this 10 years of collaboration, my colleagues that are here watching, and my family. I mean, everyone that I know that somehow collaborated and helped me to be here. So thank you. Oh gosh, what do I do now? Stop sharing here.
Okay, thank you, Elisabeth. During your talk, we had many comments, but not really a question. If anyone wants to make a question, we still have 7 minutes. I will just read some of the comments, okay?
Okay.
One comment from Francesca. A great analysis, Elisabeth. I think a lot of the challenges you mentioned in relation to taxonomy apply largely to other disciplines, especially disciplines where a large amount of observational data is required. So what do you think about it's similar the experience you have as a research in taxonomy in other disciplines?
Well, I think that those challenges actually apply to all fields of basic science, because working with basic science is, I remember that once a teacher told us that working with basic science is like you are doing research with a wall just behind your eyes and you cannot see what's beyond this wall. So you're just like producing data and sometimes you don't see the immediate applications of it. So this is why I think we should work with other institutions and NGOs and people that are aiming to translate this basic knowledge into more, I would say that a more palatable information for the public, because funding for basic science is mostly done by government. So if the society does not understand the importance of what we're doing, we'll probably be devastated.
Okay, we have another comment now from Carla. I agree. Great advice from Elisabeth. For ECRs to pursue international funding collaboration experience. Opens many doors and also creates global attention for our countries. You can talk a little bit more on that, Elisabeth, about what do you think about that? How getting this international grant change your PhD?
Yeah, so 2 experiences that changed my PhD would be this grant by Nigeria and also the exchange program, because that's what I just said, like not only the NGO translates your work to the wide public, but also, as Carlos said, it creates global attention for our country. So we are I mean, we are very lucky to live where the biodiversity is, although we don't live where the funding is. So this is why sometimes we gotta seek for international funding and many other ways of collaborating, because people are actually interested in what's going on in the Neotropics and the Afrotropics. So this will definitely draw attention. And I hope that it turns out into opportunities for local scientists. That's what I think the most important, like create opportunities for us, for Latin people and for African people to work in their own countries, in the places where we live.
Great. We have one question from Angus Patterson, that is the main director of SAIAB. Elisabeth, as the director of the Institute, what do you think are the 3 most important things I should be doing to improve the lives of postgrad students?
Wow, this is a difficult question, but I think that, for example, I will take as an example my grad school here in Brazil, that I think all of these chances I had were facilitated not only by my advisor, my collaborations, but also by the program. So we have, okay, 3 on. So we have like a really open channel with the director. So he's always available to help us, to talk to us, and to understand what is going on. So they listen to students. And second, I think that integrating students is also really nice. And third, making students know what opportunities are available. So if you are aware of a grant opportunity and exchange opportunity, to share with these people and to help students to achieve their goals. In that sense, that's what I would say. I think that I had 3 again.
Okay, let's see if there are any other question. Yeah, I think no question. We still have like 2 minutes.
So I can see here the comments on the chat, and thank you all again. This was a really nice experience.
Okay, great. I will now just wait for Ryan if I should just start introducing our next speaker.
So I'll turn off my camera and stay here to watch the next lecture.
Yes, if a question come, you come back, but I think not. I think I will proceed. I will keep going. Okay. So after this talk, I will introduce for our next and final guest speaker. That's Melanie Stiassny, one of the leading researchers on ichthyology, specialist in African fishes. She's curator of the American Museum of Natural History in the fish collection. That's one of the biggest in the world related to African fishes, and will be super interesting to listen about her research in the Congo, a really interesting research that focus on taxonomy systematics, but also evolution, on the formation of the lower Congo and how it affected the fish morphology, the diversification and species flock. So it'll be certainly a really interesting talk. So that's it. Melanie, thank you for accepting this invitation. You can start.
Sure. Okay, thanks, Pedro. Thanks, everyone. I'm going to share my screen. So here we go. Let's hope it works. How's that looking?
That's great.
Yeah.
Is that great? Good. Well, Elisabeth, fantastic talk, and actually so nice to see you again, even if it is only on Zoom. So I'm going to be talking today, and I want to thank you, first of all, for inviting me to talk a little bit about my work in the Congo. So this is going to be a little bit different from Elisabeth's excellent talk. This is going to be much more just about the work that me and my team have been doing. So thanks very much for this invitation. Okay, now before I get down to this, I want to stress that everything I'm going to be talking about has been a collaborative effort. And nothing in science gets done alone. And I know that you will all agree with that. it takes a team, and the best science is collaborative science. And over the years, I've had the honor of working with a fantastic team of co-workers. Now, my studies began in this region in 2006. So yeah, probably before some of you were born. No, a long time ago. And since then, I've been working with a great team of Congolese ichthyologists. And here are some of the core members. Now, I'm so proud of this team, because now I'm beginning to see them reach out to their own students to introduce them to the delights of Congolese ichthyology. That's been tremendously rewarding for me, and really very much of the core goal of my work in the Congo. Okay, now, of course, many others have contributed to this project, and over the years, there have been far too many who've worked with me for me to name them or even picture them. But here are just a few of the current team to give you a sense of how this is a group effort. Okay, I said I've been working in Central Congo since 2006, so there's a lot for me to say. And given the time slot that I have, I'm going to really condense this down. And I'm just going to point to some of the newer methods that we've been using to study this system. And I'm also going to touch briefly on some of the results and some of the highlights of the work. Now, I'm going to be focusing on a small corner of the Congo Basin, the lower Congo River. I'm probably going to refer to this as the LCR or the lower Congo. It's just whatever's going to come out. And here it is. The LCR represents the final course of the main stem of the Congo River as it flows from Port-Malabo to the Atlantic Ocean. It's very short. It's just close to about 350 kilometers, and it represents less than 2% of the area of the entire Congo Basin. So it's a tiny portion. Now, at the time that the Congo reaches Port-Malabo, it's drained some 3.7 million square kilometers of Central Africa, and its average rate of discharge is over 46,000 cubic meters a second. Now, just to give you a slight comparison, that's twice the discharge of the largest river in the United States. That's the Mississippi River. It's twice the discharge of the Mississippi as the Mississippi flows into the Gulf of Mexico. Now, this massive volume of water flows down a narrow, bedrock-constrained, rock-strewn channel on a contorted route to the Atlantic. And by rock-strewn, I'm not exaggerating. I mean, here are some typical views of the lower Congo, kinds of fish habitats that we're talking about. Perhaps most critically, it does this while dropping some 280 meters in elevation. So over a distance of 350 kilometers, it drops 280 meters in elevation. And the result, of course, is a series of high energy rapids. And I'm just going to show you a very short clip of one of these rapids to give you a sense of the size and the power. And just remind yourselves, you're not looking at the ocean, you're looking at the lower Congo River. Okay. Oh no. Sorry. How am I going to move this on? Oh, sorry about that, folks. Okay. Now, one of the first things when we started work on the Lower Congo we needed to do was to actually map the location of these rapids. Now, mapping their locations turned out to be quite a challenge because, as you see from this slide, for most of its length, the Lower Congo flows through a series of deep gorges, and there's very, very little road access to the river itself. And even when you manage to get to the river, it's essentially non-navigable for most of its length due to the rapids. So to really get at this, we needed to rely on reconstruction of remotely sent satellite data. And we used the differences in reflectance as surrogates for different habitat types. Now, at this point, we were concerned with mapping fully cross-channel rapids, so rapids that completely spanned the channel. because we suspected that it was these rapids that are forming dispersal barriers between populations of fish along the river. And now here's an example of one of these cross-channel rapids that we identified from satellite data. And you can see here the habitat in light blue. Sorry, that's my alarm clock going. It's quite early in the morning here. The coloration in light blue represents whitewater or rapids. And you can see it completely blocks the channel at this point. Now we compiled all of this data together. And here at a very broad scale is our mapping of the location of each of these major cross-channel rapids on the Lower Congo. Okay, so much of my work as a museum curator has been geared towards trying to reduce what we call the knowledge deficit side of biodiversity studies, biodiversity discovery, biodiversity documentation. Now, I know that descriptive taxonomy and biogeographic baselines are not always the most gripping topics for a seminar, but I do want to stress, and I think Elisabeth has done this also, but I do want to stress the fundamental importance of basic taxonomic and systematic knowledge for all evolutionary and ecological studies, or biological studies in short. I mean, after all, what is the most important thing you need to establish when you're confronted with a jigsaw puzzle? I mean, be it a food web or a community or an ecosystem. What's the first thing you need to establish? Well, you need to know what the pieces are, and you need to know that you've got them all. Basic taxonomy and systematics. So let's look at the taxonomic level first. And the first study to show that the Lower Congo is, for fishers at least, an interesting place was this one published in 1976. Now, in this short stretch of river, Robertson Stewart reported finding 129 species, which is a lot, but more importantly, or more surprisingly, they also found that 34 of those species were considered to be endemic to the system. Now, this high level of endemism is noteworthy for an African river, and particularly for such a short stretch of river. But here's the jigsaw puzzle issue. Since our work began in 2006, we've recorded more than 330 species, of which more than 90 are endemic to the Lower Congo. In other words, we've shown that in just 2% of the area of the entire Congo Basin, we have represented almost a third of all Congo Basin fishes, and almost a third of those are endemic to the Lower Congo. Now, this is truly noteworthy, and it makes this system, the Lower Congo, truly a globally outstanding hotspot for aquatic biodiversity. Now, what is it about this system that makes it such a locus for fish diversity? Now, that's what I've been working on since 2006, and believe me, it's a long story, and I'll get to as much of it as I can later. But first, let's very quickly look at some of those species I'm talking about to get more of an idea about why careful taxonomic work is a key baseline for all studies. So here are some results from a study where we were looking at the cichlid genus Teleogramma. In this study, we sequenced genome-wide SNP data to investigate essentially population structure in relation to rapids. And we did this here, concentrating on the first main cross-channel rapid outside of the pool. And here it is. Now, I won't go into detail, but the bottom line is that despite the very short distance, at 1.25 kilometers, separating these cross-channel populations, differentiation was extraordinarily high between them, and especially when you compare it to the differentiation among populations over a much longer distance on just one side of the bank. So this was interesting, but later, careful morphological analysis, and now in this case, using a combination of 3D geometric morphometrics based on CT scans of neurocrania, and using some very cool software that we can look at CT scan data, and we can actually analyze bone density differences across various structures. And using these new techniques, we were able to show that what we assumed to be multiple populations of a single species in fact represents two distinct taxonomic entities, two species, one with a highly restricted distribution and the other which is much more widespread. Phylogenetic analysis of the same data confirmed that, yeah, we have two species. They're reciprocally monophyletic, they're sister species. So now, of course, we must revise our taxonomy to reflect the true situation in our study system so we can begin to look at what's really happening. Now, we have many such examples where analysis of molecular data and morphological data has refined our understanding of a complex system. Knowing the pieces of the puzzle is critical, and far from being outdated and old school, basic taxonomy and the understanding of phylogenetic relationships really is key to gaining a full understanding of any biological system. So I just want to make that point forcefully. Okay, now we've documented numerous examples of longitudinal structuring along the Lower Congo associated with the location of those major rapids. Now, I'm sorry, this graphic is a bit unclear. It shows longitudinal structuring here between species, and up here it's showing structuring of populations within a species in response to some of these hydrological features, primarily rapids. I'm not going to go into detail, but again, the bottom line is that we do have tons of evidence that rapids do form barriers to varying degrees of efficiency, but they do form barriers, and that they've likely acted as the promoters of speciation in the past and are continuing to promote diversification within species today. Now, that rapids form barriers makes perfect sense, and the LCR story could end there. You know, we've explained it, but it turns out things are much more complicated. We were beginning to find instances of population divergence across sections of the LCR where there were no rapids at all. There was no apparent barriers. There was no seeming reason why we should be seeing populations separated over short distances within the absence of any obvious barriers. And I'm going to get to that to a minute. But first, I want to point out that most of these new species that we've been discovering in the Lower Congo and documenting, they're pretty normal in the sense that they're typical members of the fish families to which they belong. You know, like these cichlids. I mean, they're elongate and they've got features that indicate that they may be somewhat adapted to rapids habitats, but they're clearly cichlids. I mean, there's nothing hugely unusual about them. And that's been the case for most of the things we've been discovering. But that's not the case for all of them. Like this very weird, blind, depigmented cichlid, Lamprologus lethops. You can hear a lot about that guy in a minute. But then there are also these. All of these are unique endemics to the Lower Congo River. They've all lost or greatly reduced the image forming capabilities of their eyes, and we're calling that cryptophthalmia. All are depigmented, and despite very different shapes and sizes, they've all converged on a set of, superficially at least, strikingly similar phenotypes. This is perhaps the most extreme example of morphological convergence across phylogenetically hugely disparate lineages. I mean, we're looking at representatives from across the tree of life of Telios here, yet they've all converged and they're all found in a single system. So this really is a pretty cool, super cool system. But I just don't have time to go into all of the details of our work. But I do want to point out that we've been using a methodology that I think is quite interesting. We've been used, because you can't compare cichlids, for example, in this example, it's very difficult to compare cichlids with mastacembalids. Their anatomy is so very, very different. So what we've been trying to do is make these couplet comparisons. So here we have the blind form of a Lower Congo endemic, and here we have a sighted form, its sister species. Here we have a blind form, the Mastacembelus bouchardi, and here we have its cis-sighted sister species, also a Lower Congo endemic, Mastacembelus brachyrhinus. And what I'm showing you here is reconstructions that I've made of CT scans that have been differentially stained with a high contrast material which allows us to visualize soft tissue. So in this example, you can see I've reconstructed their brains, and I've reconstructed some of the associated sensory structures, the olfactory rosettes, the eyes, obviously, the optic chiasma, the otoliths, etc. So even though the anatomy of these two groups is completely different, by comparing these couplets, certain generalities become visible. And I'll very quickly mention some of these. For example, some are completely expected and make perfect sense. In the blind forms, the otoliths tend to be much larger and better, more heavily, densely ossified, and more structurally solid. So in the blind forms, you see this compared to their sighted sister species. That makes perfect sense. Another thing that kind of makes sense is if we look at the cerebellum. If we look at the cerebellum of the sighted species, in both cases, the cerebellum is more discrete. It's a better formed structure. And that makes perfect sense, because if you've got eyes and you're navigating a three-dimensional world, the coordination center for all of that is the cerebellum. So that makes sense. And then we found some other things that don't seem to make any sense at all. Is it seems that in the blind Lower Congo endemics, the olfactory rosettes are disorganized and poorly developed compared to the eyed congeners. So why, if you can't see, you can't smell very well, I don't know. But these are the kinds of things that we've been looking for, so that we can characterize this remarkable example of convergence. Anyway, this is ongoing work, and I got to move on because time's running out. Okay, I want to finish with one last piece of the puzzle, and this gets back to that question, or that puzzle, where we were finding divergence in the absence of rapids. And it all began with that blind cichlid, Lamprologus lithops. Now, lithops has been found only in a very small area of the Lower Congo. It's about halfway down the Lower Congo, around a small village settlement called Bulu. The fish is the only known blind cichlid, but what's really much more unusual about this fish is that it's only ever been found dead or dying at the water surface, which is surely one of the most unusual situations for any species. It's known only from dead or dying exemplars. Now, over the years, we've gathered numerous specimens of lithops, always dead, always from this same small region of the Lower Congo. Now, on one trip, some local fishers brought us one specimen of lithops that they'd found, again, at the water surface at the bank. But it was just alive. And as it died, I was holding it in my hand, as it died, we actually saw bubbles form slowly under its skin and over its gills. So it seemed to fill with air. It really did appear to be suffering from catastrophic decompression syndrome, which is the fish's equivalent of the bends. So, sorry, let me go back. So up until now, oh gosh, I'm so sorry. Let me go forward. Okay, so up until then, up until that point, our work had concentrated on the rapids, which are shallow water phenomena. Now, for the first time, the question was raised, could there be deep water in the Lower Congo? And now, it's a very long story as to how we finally resolved this, but very briefly, with colleagues from the US Geological Survey, we've deployed this wonderful piece of equipment, this acoustic Doppler current profiler. And with this setup, we were actually able not only to measure what the bottom of the channel looked like and the depth, but we were also able to visualize the flow dynamics in that channel throughout the water column. So here are some of the results. And again, I know it's a bit complicated, but I'll quickly rush through it. What you're looking at here is a cross section through the channel at this point, at bend 2. And at that point, that's the region where most of the dead lithops were found. Now, the first thing I want you to look at is the depth. The bottom of the channel at this point is around 160 meters in depth. Now, that's by far the deepest point in any river ever recorded. And incidentally, it's not even the deepest place in the Lower Congo. So yes, there is a lot of very deep water in the Lower Congo, as well as very shallow rapids. But let's have a quick look at the water currents. And if I can explain very briefly, this is color-coded to show you the vertical velocities of water flow. And the orange and the reds, the orange and the yellows are going directly upwards, and the blues and the greens are coming directly downwards. So what we see are these persistent vortices, persistent cells of upwelling and downwelling water. And in between them, there are these vertical jets here in red, which are just shooting up to the surface, rapidly transporting water from the benthos all the way up to the surface, some 160 meters above. And you can just get a little sense of the scale of all of this if you look at this inset. This is what these boils and suckholes above one of these big canyons looks like. And to give you a little idea of scale, these are actually 2 kayakers. So you can see how enormous these structures are. So we hypothesize that our little Lamprologus lithops is living close to or on the bottom. And we've got lots of anatomical evidence I can't go into here, but that suggests that this is a benthic dwelling fish. And our idea is that it's living in the bottom in a kind of safe zone. Every now and again, individuals will get entrained in one of these jets and will be swept up to the surface, dying of catastrophic decompression on the way up. And that's why we're finding them dead. Now, more of that in a minute, but let me just quickly show you another transect, because I think this one really says a lot about the story of the Lower Congo. Now here, we're looking at a cross section through the river at Bend 1. Now at Bend 1, the river looks completely normal. It's completely flat, doesn't seem to have anything going on at all. But if we look at this ADCP output, in this time, we're looking at streamwise velocities. So the reds and the yellows and the oranges are traveling very fast towards you. You're standing in the channel looking up. It's coming downstream very fast. The blues and the greens on either side are going in the opposite direction. So you have this central core of river coming down towards you, and then you have at the sides, the 2 shorelines have slower flowing water actually backing up, going in the opposite direction. So really, basically what we're looking at here, and look, this is over a very short distance. We're talking about a distance of about ½ a kilometer here. So this shoreline is completely separated from this shoreline by the sheer zone of flow separation. If you're a little fish living here, there's no way this could be 100 miles away. It's ½ a kilometer away, but it could be 100 miles away, because there's no way you're going to be able to swim across there without being flushed down. Basically, we're looking at rivers within rivers. And okay, so even in the absence of rapids, it seems like the waters of the Lower Congo really appear to be promoting separation of populations. Now, the Lower Congo is clearly an unusual river system, and we can talk about all the ways in which it's unusual. It's rocky shorelines with shallow rapids, often immediately adjacent to phenomenally deep holes and canyons. And that combined with the vast amount of water that's flowing down this narrow channel results in a complex array of extreme flow regimes. So in short, our findings suggest that the river itself may be the powerful driver of micro-allopatric isolation of populations. And we're seeing this often over remarkably small geographical scales. Now, acting over time, clearly such a system will generate a lot of diversity. So this really is a core system, and I'm still there, and we're still turning up new things. And I'm going to end very, very quickly by just turning to another component of the studies that we're doing in the Lower Congo. And this is gene sequencing and genome interrogation. Now, we've started with lithops, and we've sequenced its genome, and we've sequenced the genomes of related lamprologine species. And again, I don't have time to go into the details, but I think we've already finding some interesting things, particularly in relation to what I've been talking about. Because remember that with lithops, we were making a guess about its habitat and its ecology, where it was living, what it was doing. Based on its appearance and how we only found it dead and dying of decompression in the region of a very deep canyon, we hypothesized that lithops lives in a deep canyon in a lightless environment. But we've no real proof of that. I mean, we can't go down to the bottom of that canyon. It would be easier to go to the bottom of the Marianas Trench than to get to the bottom of that canyon at Bulu. So we can't make direct observations. Well, however, in analyzing our genomes, and we weren't looking for this, but this came out, in analyzing the genome of lithops in comparison with the normal eyed forms, we found that many genes implicated in the repair of UV light damage to DNA, in lithops, many of those genes are broken. They've lost their function. Now, that's not to say that lithops might not have other ways to repair its DNA, but this seems to us to be very strong supporting evidence that lithops is living very deep in a lightless environment where it never gets exposed to UV light. So selection pressure is relaxed. There's no need to maintain an elaborate UV repair mechanism. And that's exactly what we see. Another example is this one. We found that lithops shows numerous mutations related to the lack of appetite suppression. I think I have this, but anyway. One interesting example is spexin. Now, spexin suppresses appetite, and its loss results in binge eating. As a result, organisms like lithops that have lost or downregulated spexin tend to be very, very fat and very starvation resistant. And in the CT scan, you can see here we have our lithops compared to a surface dwelling congener, and it's full of fat globules. It really is fat. And it's interesting that we did CT scans of the famous blind Mexican cavefish, Stiassny mexicanus, and here she is, full of fat globules. And here's her surface dwelling. In this case, it's a cave, not a deep canyon. But here's her surface dwelling member of actually the same species, completely lacking those fat globules. Interestingly, it's a different gene that underlies this than in this, but that's, those are details. Anyway, the loss of function of spexin makes perfect sense if lithops lives, as we hypothesize, at the bottom of a deep canyon where food is only sporadically available. It makes perfect sense to binge eat like crazy when food is there, and then rely on these stored fat reserves when it's not. Okay, so our genome studies are really only just beginning, and there's clearly a lot of work to be done. But I think the results are already kind of interesting and promising. Okay, I've run out of time and I'm running out of steam, so I'm going to wind down there. Obviously, there's still a lot of work to be done, but I hope you'll agree that this whiz through review of our work in the Lower Congo, we've shown it really is an extraordinary place. And by integrating hydrological, morphological, phylogenetic, taxonomic, and genomic components, we hope that it's going to become a real model system to begin to address a whole set of fundamental questions relating to diversification and adaptation in freshwater systems. And with that, I'm going to stop, but I can't end without acknowledging the people living alongside this amazing river. They know more about the fishes living there than I ever will. And our project simply couldn't have happened without their expertise and help. And finally, my gratitude to all the fish that gave their lives so I could give this talk today. So with that, I'm going to shut up and thank you once again for inviting me to give this presentation. And what a lovely event it is. And I wish I could be there in person. Okay, thanks. Pedro?
Yes, thank you a lot for the talk. We have quite some questions here. Let's go. the first question was from Francesca. Melanie, do you have an early, what do you have an early stage connectivity pattern to confirm, reject the rapid barrier corridor mechanisms? Do you have any early stage connectivity patterns to confirm, reject the rapid barrier corridor mechanisms?
Well, yeah, I mean, yes. I think if what you're talking about is that longitudinal structuring, and I showed that graphic, which I said was kind of complicated and too complicated to go into. I mean, that analysis, particularly the analysis within of population response to rapids within a species. Yeah, we've used all sorts of modeling to look at the directionality of migration and the strength of migration and the expectation for isolation by distance and all of that. So yeah, and the picture is actually quite complicated. It's not simple. Not all fish respond to the rapids, the same rapids in the same way. So it's clearly very complicated. But as a generality, it's true. It holds. The rapids are dividing up populations. I hope that, Francesca, answers your question.
Okay, I have another one now from Angus. So man-made changes in flow, abstractions, dam and channel mining, which change rapids, size, energy will disrupt evolutionary processes. And also in this question, if you have any knowledge about maybe a dam that they're planning to construct.
Yeah. This is the perennial issue. And yes, I think our work shows very, very clearly that damming is going to really, really change this whole system. So yes, there are major plans. And in fact, I get contacted nearly every day recently by companies that are putting in bids to work with Fortescue, Fortune Industries, who want to build dams all the way down the Lower Congo. So yeah, it's definitely been revived at the moment. It's being presented as green energy. They want to produce hydrogen using this energy. The IUC and the latest COP conference had a motion actually to really strongly oppose the development of more dams on the Lower Congo. So hopefully, that's going to have some impact. But yes, you've absolutely nailed it. The system it's the river itself that's producing and maintaining the diversity in the system. So it's the worst place in the world to put more dams.
Thank you, Melanie. Yeah, that's a pity. Let's hope they don't build any dam there. Another question from Francesca. Does river flow in calmer water present the formation of fronts analogous to the coastal waters? What could influence barriers locally in terms of space or time?
Sorry, where would I find that question? Because I can't.
In the chat, you can click in the chat.
River flow. Does the river flow in calmer waters present the formation of fronts analogous that could influence barriers locally in terms of space and time? Yipes! I don't know. I don't think so. I mean, what we were seeing was zones of shear flow separation. If you want to call that a front, maybe, I don't know. Yeah, I'm sorry, Francesca. I don't know the answer.
Melanie, we have an interesting question here from Monica. That was one of the guest speakers today. The SPX genome aspect. Have you observed this in the species that has similar characteristics to letops based on convergent evolution?
Fantastic question. And that's one of the things that we're doing now is we're starting to sequence the genomes of all of those other cryptophthalmic forms and to look for exactly these sorts of things. And in fact, we're even extending it out to Elisabeth and Pedro would be interested to this with gymnotiforms in South America. So yeah, we're definitely looking for that. But at the moment, no, we don't. But I will say one thing, and this makes the whole question so complicated. As far as we can see, lithops is the only one of these cryptophthalmic forms that's actually living in depth. It's the only one that we only ever find dead. It's the only one that has these fat reserves that we just serendipitously stumbled across when we were looking at CT scans. It's the only one that has that. The others don't. And the others, we find them alive and we found them close to the surface. Now, they may be living at depth, but they're capable of coming up and down in a way that lithops isn't. So personally, I wouldn't anticipate that we will find that at SPECS or a similar appetite suppressant control in these other things, but maybe, I don't know, maybe it's more likely in some of the, like the Astyanax, the cavefish, maybe in some of the gymnotiforms. But yeah, so no, but that's what we're doing. We're sequencing all of these guys.
Melanie, I have a question.
Sorry, we did it with cichlids because obviously the genomic resources for cichlids are so much richer than, say, Stomata rhinus and mormyrid. We're going to have to do a lot more work to get that genome put together than the cichlids. Yeah, sorry.
Yeah, I was waiting a question. Now I have a question, but I have a quick question before the one I will read. Is why the Lower Congo in 2006? It was a blind shot, or you had some knowledge through collection, looking, you only choose probably Lower Congo will be the best place to do research.
Yeah. Oh, that's a great question. I wish I could claim some, prescience, but basically I'd worked with my very dear colleague from the Belgian Museum in Emmerich, Guy Tugels, and we worked together on a book of the fishes of Central West Africa. So basically, Cameroon, Gabon, coastal Congo. So when that was done, we thought, well, now we're going to take on the Congo. And we kind of realized you couldn't do it all in one chunk. So we said, well, let's start with the Lower Congo, and then we'll do that. We'll do that in a couple of years, and then we'll move on up. But I got stuck in the Lower Congo. So I'm very glad we started with the Lower Congo, not the Upper Congo. So it was kind of chance, but worked out well.
Okay, thank you for your reply. And now a question from Albert Chacona. What methods do you use to sample these turbulent habitats?
Yeah, it's very, very hard. And I can tell you, if you remember looking at some of those rocky habitats, it's like looking for a needle in a haystack. I mean, we've got 330 species documented. There's so many more. There's so many more. Every time we go, we find new species, because it is just stochastic where you are, what happens to be there at the moment that you manage to catch it. It's extremely difficult sampling in the rapids, obviously. And we rely a lot on, local fishing expertise, cast nets, dip nets, hooks and lines. But to really get at the meat and potatoes of it, with permission, we do use the ichthyoside rotenone. And by strategically placing rotenone in the stream that's flowing over a particular area, and then standing at the other side of that area and catching everything that's fleeing out of it, that's basically how we get the better sampling. Okay,
thank you. And that's it. We have no more questions. We have 1 minute.
Wow, good timing.
Yeah, good timing. Okay.
Great. Well, then I'll just say thank you so much. It was a pleasure to have a chance to chat with you, and I'll be turning off my camera and I'll be sitting here watching what's going on. I mean, listening to what's going on. Thank you.
Okay, thank you, Melanie. And now I will leave too, and the new chairperson will lead now. I just need to check who is. Just a second. It'll be Dumi.
Thank you, Pedro.
Okay, thank you, man. Good.
It was a very nice session. Okay, we'll just wait a bit for the people who will be going to the next session to leave, and then we'll start with some very nice presentations. So we'll be having 2 MScs and 4 PhDs. We'll start exactly in like now. So our next speaker will be Hima. She'll be telling us about the work from Lake Nyasa. Very good, Hima.
Hello everyone, my name is Hima Rama. Today I will be talking about using baited remote underwater video systems to monitor a small, commercially important cichlid species in Lake Nyasa. Lake Nyasa, also known as Lake Malawi, is a transboundary lake which is a critical shared resource for 3 developing countries, Malawi, Mozambique and Tanzania. It is a well-known biodiversity hotspot and is home to the largest recorded number of freshwater fish species in the world, bulk of which are endemic to the lake. The lake currently supports a number of industrial and artisanal fisheries which are a critical source of nutrition for local communities and supports the livelihoods of more than 1.6 million people. It is an attractive destination for ecotourism because of its incredible biodiversity. Live fish specimens are also collected from the lake for sale in the international aquarium trade. Therefore, the lake is heavily relied upon by surrounding communities both ecologically and socio-economically. However, human activity on the lake, coupled with an increasing population in surrounding communities, has resulted in fish population declines as a result of overfishing, destructive fishing methods, and habitat degradation. In order to alleviate these impacts, the management of these areas is critical and needs to be underpinned by frequent and long-term monitoring of species populations. This needs to be done while also minimizing financial strain on the surrounding countries. The use of baited remote underwater video systems, or BRUVs, is a cost-effective, non-invasive management tool that has proved useful for monitoring marine fish communities and larger fish species within Lake Nyasa itself. The aim of my research was to see if BRUVs could be used to extract critical population information about abundance and habitat preferences of a smaller fish species in the lake. BRUVs were deployed at various different points within Minnows Reef, an area in which the study species Metroclima istere are most commonly caught for sale in the aquarium trade. Metroclima istere is a sexually dimorphic species, with females being bright yellow in color and males a light blue with gray vertical bars. It is one of the most commonly kept African cichlids by fish keepers in the hobby. The footage collected by the BRUVs was analyzed to determine the habitat preferences of the species by seeing which habitat variables were associated with a higher occurrence of individuals. We found that the main driver of Metroclima istere abundance was substrate. A principal component analysis showed that sites with both male and female individuals present are correlated with rock and cobble. In comparison, sites with no individuals present correlated with sites dominated by sand and vegetation. Therefore, the species as a whole is more abundant in rocky habitats. However, we also found that males and females exploited slightly different niches. Sites where males alone were present were common across all habitat types, whereas females showed a clear preference for rocky habitat. Bulk of the sites where the species was not sighted were dominated by sand. In comparison, there was only a small proportion of sites in rocky habitat where the species was absent. The results showed that Metroclima istere have a clear preference for rocky substrate. We think that this is because they are brightly colored and require the hiding spaces provided by the rock to avoid predators. Furthermore, the primary diet of the species is the algae which grows on the surface of these rocks. Because the bright yellow females are more conspicuous, they are probably more dependent on the refugia provided by rock, whereas the male's light blue color is far more likely to blend in against the sand, so they have a lower risk to predation. This allows them to disperse further than the females, which is necessary because the males of the species exhibit territoriality. In conclusion, BRUVs were effective in collecting critical population data on Metroclima istere, and could be implemented to monitor the smaller species in the lake to underpin management. Analysis of BRUV's footage revealed that protecting or rehabilitating rocky areas could be useful for preventing a population decline for the species. This means that BRUVs are a viable management tool for the species and others similar to it. The data provided by the method is unbiased towards fishery species and can be useful for the long-term monitoring of the overall fish communities in Lake Nyasa. Thank you.
Thank you, Hima, for showing us the importance of videos on monitoring habitat systems. So the next speaker will be Nawa, telling us about the influence of Chirex, the crayfish in the Zambezi system. Over to you, Nawa.
Hello, everyone. I would like to welcome you to my presentation. My name is Nawa. I'm a PhD student at Rhodes University under the Department of Zoology and Entomology. I'm being supervised by Professor Ryan Wasserman. The title of my presentation today is Assessing Effect of Non-Native Red Crow Crayfish, also known as Chirex quadricarinatus, on the Barossa floodplain fishery. Invasive alien species have been found to cost economies around the world billions of dollars. These costs incurred come mainly in form of ecological and socio-economic impact as well as associated management. Cost assessment of invasive alien species has mainly been conducted in North America, Asia, and Europe, whereas assessment in Africa remains relatively unreported. Aquatic invasive species are responsible for a large amount of the cost. For example, invasive crayfish have been found to cost about $1.3 billion per annum, but most of this cost is attributed to Europe and North America. That is despite Africa having its fair share of introduction of non-native crayfish in the past 3 decades, which has seen about 5 crayfish having established feral populations. This study, therefore, aimed at quantifying impact of non-native red crow crayfish on the Barossa floodplain fisheries. The study utilized a roving crew survey method by administering electronic-based tablet questionnaires in which fishers were interviewed on landing site to assess damage on fish as well as fishing gear. Preliminary findings show that there is high fish spoilage in low water compared to high water when the floodplain are flooded. And most of this damage comes from gill nets compared to any other gear. And this could be because crayfish has ample time to feast or feed on the fish that has been caught on the gill net. The gill net have also been found to be adversely affected in terms of damage. They're getting more damage than any other gear on the floodplain. This is because the crayfish tends to get entangled on the net, and in the process of either escaping or being removed by the fisher, they end up causing damage to the gear, and therefore the lifespan of the gear has been significantly reduced, and the fisher is therefore required to buy or get more extra money to buy new fishing gear. The cost arising from spoiled fish and damaged fishing gear has negatively affected the livelihood of fishing community on the floodplain. And as the invasion spreads across the plain, the contribution of the fishery to supporting rural livelihood and contributing to the local economy will become more compromised. It's therefore important to look at policies and management recommendation to find ways of mitigating this problem. I would like to give special thanks to my supervisor, Professor Ryan Wasserman, and also SAIAB NRIF, as well as WWF Zambia for funding my studies. We have come to the end of my presentation. Thank you for being in attendance.
Thank you, Nawa. This was a very nice presentation to highlight the influence of invasion on the fisheries. So there are some questions. You can write your questions on the chat box, and then at the end of the session, the speakers will address them. So the next speaker will be Dina Mkari. She'll be telling us about her work in the Groenvlei. Over to you, Dina.
Hi everyone, my name is Dina Mukari and I'm a master's student with SAIAB and Rhodes. I'll be presenting about the life history of non-native common carp at Groenvlei in South Africa. Carp was introduced in 1859 into South Africa for ornamental purposes, and the recent distribution map shows that this species is now present in all major water bodies of the country. Carp is also identified as one of the world's worst invaders, and its impact include increasing turbidity and nutrients in water bodies, impacts on native species such as predation and competition, and introduction of parasites and diseases. My study took place in a small isolated lake within the Gokama Nature Reserve in the Western Cape Province of South Africa. This lake is used by the locals for both recreational and subsistence fishing, and here carp was introduced illegally in the 1990s. The increasing biomass of carp at Groenvlei poses a big threat to this natural system, and due to this, there is currently ongoing efforts to reduce this biomass. Knowledge of the life history of carp at Groenvlei will aid in understanding the population dynamics and recruitment patterns, which are very important in order to optimize the current control efforts. And to do this, we studied the growth and reproduction characteristics of carp in the system. In order to do this, we started by validating the rate of annulus formation on Asteriscus otolith of carp by collaborating with an ongoing study, and from here we found that one growth annulus forms each year on the otolith of carp. The growth annulus is shown by the dark line on this picture, which I've also circled in red. We then removed otolith from 230 carp and sectioned them and counted the growth annulus under the light microscope. Each growth annulus represented a year. And then from there we plotted a graph showing the age of carp against the length. And as shown here, carp grows very fast between the ages of 0 to 5 years, reaching about the length of 400 millimeters, whereby now the growth rate slows down as more energy is now invested in reproduction. My final objective was to understand the reproductive biology of carp at Groenvlei, and this was done by physically assessing the gonadal development stages following a criteria used for carp. In general, gonads increase in size towards the spawning season, and since the carp were collected in November 2020 during the peak spawning season of carp, we were fortunate enough to see the ripe gonads for both males and females, which I managed to capture and show here. This data was used to calculate the average length at 50% maturity for both males and female carp at Groenvlei, and the females attained maturity at an average of 262 millimeters, which was about 2 to 3 years of age, and this was earlier compared to the males. Studies have shown that carp are highly fecund. Their relative fecundity ranges from 100,000 to 300,000 oocytes per kilogram of body weight. During this study, we found that common carp at Groenvlei shows traits of a successful invader. They grow very fast, they reach maturity at an early age and are long-lived. Warm temperatures and high system productivity, which are provided by the lake, together with lack of natural enemies in the systems, are the reason for their success. And the finding of the study brings us one step closer into understanding the invasive potential of carp at Groenvlei and other systems with similar environmental properties here in South Africa. I would really like to end this presentation by thanking everybody who made this project possible, starting with the financial support and my supervisors, Dr. Josie Pack, Prof. Paul Colley, and the late Prof. Olaf Vale, who gave me such an incredible opportunity. And thank you so much for listening to my talk.
Thank you, Dina, for giving such a great talk and keeping us informed on invasive species in South Africa. The next speaker will be Peter, who will be telling us about some work in Lake Harib. Over to you, Peter.
Good day, everyone. My name is Peter Swanepoel, and my topic for today is assessing small-scale fisheries potential of Lake Harib, South Africa. Okay, the introduction. A national freshwater or inland wild capture fisheries policy was approved by Parliament in August of 2021, earlier this year, and I'm going to quote directly from this policy. First part is, South African inland fisheries resources have the potential to contribute to food security, job creation and economic development. And then the second quote is, governance and management arrangements will be established to ensure that inland fisheries are ecologically, socially and economically sustainable. And an important word for this study is the word sustainable. So to develop inland fisheries on a sustainable basis, locality-specific, relevant and current information is required to make informed decisions. The objective of this current study is to present new or current information through relevant scientific research that can be used for sustainable inland fisheries development of Harib Dam. Okay, so I have already presented materials and methods at a previous SAIAB symposium. So for the purpose of this talk, I will go straight to the results of the study, and I'm going to talk about the roving creel surveys, which was done between October and September 2021, where 397 interviews were conducted with fishermen of the Harib Dam, and a total of 858 fish specimens caught by these fishermen were measured and weighed in both the Harib Dam and overseas fishing areas. So through the roving creel surveys, I have identified the users or utilizers the dam for fishing. This pie chart shows that most of the fishermen are subsistence fishermen, with more than 80%, with less than 10% of them representing subsistence fishermen. The bar graph on the right illustrates the species versus angler catches in catch per unit effort. The highest catch per unit effort was recorded for the smallmouth yellowfish, followed by the common carp. So through the roving creel surveys, 67% of all anglers interviewed had caught at least a fish, where 33% of all anglers interviewed had a zero catch. The red bar indicates the zero catch, and the yellow bar is the catch per unit effort per angler per hour. We also calculated the mean catch per unit effort for anglers of the Harib Dam, which was calculated at 0.65 kilograms per angler per hour. The graph illustrates the catch per unit effort for the sampling months, as well as the probability of an angler to have caught at least a fish at the time of interview. Both the catch per unit effort and the probability is lower in the winter months and higher in the warmer months, the summer months. This can be due to the lower metabolic rate of fish in the winter, which results in less feeding and therefore a low catch per unit effort. The duration of a fishing day was also calculated, and the average hour fished by a fisherman throughout the year was 7.09 hours per day. The graph showed that the time an angler spent fishing is relatively similar throughout the year, except in June, where the average time was lower at 6.52 hours per day. Okay, so this is the result of the fishing effort surveys. A total of 89 days of fishing effort surveys were conducted. During the surveys, all fishermen were counted twice daily. This was done in the Overstern and the Grypdam fishing areas, and the average angler per day in Grypdam was calculated at 20.9 anglers per day. The graph shows the average anglers per day during the sampling periods of the two respective open fishing areas. The number of anglers increased during May and June in Overstern, and the amount of anglers fluctuated throughout the year in the Grypdam area. Quite interestingly, the fishing effort increased in the winter months, but the catch per unit effort was the lowest during the same time. Okay, so to sum up the results of the creel and effort surveys, the mean catch per unit effort was calculated at 0.65 kilograms an angler per hour. The mean duration was calculated at 7.09 hours a day. The mean catch per unit effort times the mean duration gives you the mean expected end of day catch, which was calculated at 4.6 kilograms a day. Average anglers per day was determined at 20.9 anglers a day. The mean expected end day catch times the average anglers per day gives you a total of 96.14 kilograms a day that are being harvested by these recreational and subsistence fishermen. So the total annual catches from anglers at the Grypdam was calculated at 35.115 tons a year. Okay, so to discuss, harvest by subsistence and recreational fishermen is substantial at 35 tons a year and requires recognition if fisheries is to be developed. They also mainly harvest indigenous smallmouth yellowfish, followed by the common carp. This is my references. I would like to acknowledge the following institutions for funding and support: the University of the Free State, the Free State Department of Agriculture and Rural Development, and the South African Institute for Aquatic Biodiversity. Thank you for listening. Please feel free to ask any questions.
Thank you, Peter, for your talk. This information is very useful for the development of our inland fisheries policies. So the next speaker will be Angus, who will be telling us about the work in Lake Malawi. Over to you, Peter. Angus, I'm sorry.
Hi, everybody. So what I'd like to present today is just a brief snapshot of my master's project. I wanted to look at the potential of beta remote underwater video systems, commonly known as BRUVs, in Lake Malawi. And this was a joint project which began in 2016 with ourselves, SAIAB, WWF, and IIP. And I jumped onto it in 2018 for my master's and started to include some Malawian data. So just a brief background. Lake Malawi was an incredible study system. It's of great importance to biologists worldwide. It has incredible biodiversity, and it's also of great importance to those that make use of it every day, particularly fishers and the families it supports. So the project originally began in the Lake Nyasa Reserve on the Mozambican side of the lake, with a northern sample site, Kobwe, extending down to Meliluko. Along the way, I made a lot of great friends, as you do, and I had an amazing experience. Many a good sunset. On the Malawian side, what we tried to achieve was getting comparable sample sites during my masters. So we looked at closed areas such as Cape Maclear to compare to some of the reserve sites in the Lake Nyasa Reserve, and some heavily fished sites such as Mkudzi Bay, just to draw some comparisons across the lake. So we speak of innovation. One of the first challenges was obviously taking the marine remote imagery platforms, BRUVs, and standard operating procedures, and we wanted to try and, leverage the platform as one does, but trying to make some changes to make the system itself a little bit more operable in the field, and adjust some of the standard operating procedures that are used in the marine realm and make them more apt for freshwater. So one of these, I guess to begin with, was bait. So we ended up going for usipa, which is a pelagic cyprinid, likened to a sardine that they would use in the marine realm. So I broke up this presentation into sort of two impact areas. So I just had research impact and societal impact. So research impact, what we wanted to do was basically draw up some of the first standard operating procedures and a way forward, or a roadmap for using stereobruvs in an African freshwater body. So the first thing you look at is deployment time. How long should a bruv stay down on the lake bed? And whether or not it's actually feasible to start a monitoring project, say over 10 years using bruvs. And both of these sort of held up. We accumulated our key species within about 15 minutes of film, which is a very manageable time frame. And our sample size over 10 years to detect a change in abundance was also somewhat achievable. This is a graph looking at the ability to detect a change in chumbo abundance over a period of 10 years. Interesting about this graph is often when you have high fished or more fished areas, you have lower abundances, so you require more samples in order to detect a change in abundance. That's what we see here in Malawi, which is somewhat more fish than what you see in Mozambique, which is an interesting result. I then did the same basically for all of the key species. And what we're also interested in looking at was whether or not the browsers were able to detect changes in abundance, looking at key factors for species. So when you look at this figure, we're looking at chumbo. As you can see, there's an effect on depth, habitat type, closure, and I guess year on the abundance of the chumbo species. Then we also wanted to actually incorporate some of the length data that you can generate from stereobruvs. So stereo, as the name implies, allows the observer to not only count fish, but we can also measure fish underwater or remotely. And what you can see here that there are significant differences in fish size, the size classes, looking at fork length, depending on the depth ranges, the habitat usage, so structured versus sand, and a couple of the different areas. So what you see here is like a somewhat classic image where you have closed areas having a lot more larger individuals, which you don't see present in the heavily fished areas, which is somewhat comparable to the catch data we were able to gain from both Malawi and Mozambique. Then when I look at societal impact, we had an amazing time while we were there. We were able to not only demonstrate our footage to the local fishing community, it was an amazing experience to see them seeing them respond to the first time seeing underwater video footage of their own lake, which was amazing. The data fed into one of the first delineations of a key biodiversity area in Lake Nyasa, which is a fantastic experience. And then we've had multiple presentations with WWF and key stakeholders trying to garner more funding and trying to get some more attention on the project itself. So yeah, this was an amazing experience all around. Yeah, and then yeah, thank you to everyone involved. Yeah, cheers.
Oh, thank you, Angus, for such a good presentation on how technology can be utilized to assess our fisheries. So our next speaker, the last one will be Phakama. Remember, we will be voting at the end of this session, so you can also post your questions, as Brian has mentioned. Over to you, Phakama.
Greetings, everyone. I'm Phakama Noto, and I'll be presenting the necessary function of marine, near-shore, and estuarine venting habitats for demersal fish in Algoa Bay. Estuaries play a critical role in the early life history of marine fish, including many economically and ecologically important species, with research throughout the world showing that estuarine fish communities are dominated by the juveniles of marine species, which make them important nursery grounds. Marine nearshore environments also serve as necessary areas for a wide variety of marine fishes, including species common to both adjacent estuarine and offshore areas. However, less research has focused on the necessary function of the marine nearshore environment compared to the estuaries, and this research gap is apparent in South Africa. Now, to fully understand the functional significance of both environments alone and relative to each other, as settlement in necessary areas, it is therefore important to concurrently study different life stages of fish from the head of the estuary to the adjacent marine nearshore environment. Species occupying these coastal areas have complex life histories as they require numerous habitats to complete their life cycles. For example, marine estuarine-associated species found in Breeded Sea. Then when the pre-fishing larval stage is complete, fish movement becomes active with some individuals entering the surf zone, which may be used as an interim necessary area before migrating into their estuarine necessary areas. Identifying core necessary areas and other essential fish habitats for exploited species is important for marine spatial planning and the development of effective management strategies. The aim of this study was to determine the relative roles of the soft bottom, benthic marine nearshore and estuarine environments as settlement in necessary areas for demersal fishes, and the objective was to determine fish abundance by life stage in these two environments, as well as identify essential fish habitats, that is the density hotspots, along the estuary ocean gradient. This study was conducted in Algoa Bay, whereby sites ranging from 5 to 10 meters depth within the marine environment were sampled. The adjacent estuaries that enter the bay, which is Swartkops and Sundays estuary, were also sampled, with 5 distributed throughout the system to cover the lower reaches, the middle and the upper reaches of both estuaries. The beam trawl net was used for fish collection, and all the species that were caught were identified in the field, and for those that we put in ID were kept for DNA barcoding. These are some of the species that we identified using DNA barcoding. Although both estuarine and marine nearshore environments were dominated by early life history stages, the overall abundance of settlement stage individuals and juveniles was 5 to 6 times higher in the estuarine environment in all the seasons. This shows that soft bottom benthic estuarine environments are far more productive necessary areas for demersal species when compared to soft bottom benthic nearshore marine environments. The settlement and necessary habitats also differed by estuarine association category, with the nearshore marine environment dominated by marine species as well as marine estuarine opportunists, and the core settlement and necessary areas for these species were located close to estuary mouths, freshwater outlets, and harbors. In contrast, the estuarine environment was dominated by marine estuarine-dependent species and estuarine species, with the upper reaches of the Swartkops estuary and the lower reaches of the Sundays estuary identified as important settlements and necessary areas for these species. There was high degree of overlap in the necessary habitat used by many lionfish species caught within the marine nearshore environment. The areas identified as settlement and necessary habitats for the exploited species in the Algoa Bay were the sites closest to the estuary mouth and harbors for marine species such as silver cob and red-spotted tongue sole. While in the estuarine environment, the upper reaches of the Swartkops estuary and the lower reaches of the Sundays estuary were identified as important settlement and necessary areas for marine estuarine-dependent species such as Cape Stamp-nose and dusky cob. This study identified necessary areas that should be prioritized for conservation as they were preferred by many important fisheries species reported. And it is therefore suggested that this funding should be used for spatial planning of the essential juvenile hotspots to develop effective management strategies for coastal fish populations, particularly in the light of anthropogenic activities and climate change. I would like to thank my supervisors and everybody who assisted me in the field and in the lab. Thank you so much.
Thank you, Kagama, for such a lovely presentations. So I will read some few questions, one for Hima and another one for Nawa. And then a poll will be posted now. A poll has already been posted, so you can vote. So one question for Hima is, could this habitat preference also found in other dimorphic Mbuna cichlids? This is from Tit. If you can answer that, Hima, please.
Hi, everyone. So yeah, we think that since the Mbuna are all, like a bulk of them are rock dwelling and sexually dimorphic, we think that yes, the sexual the also be applied to other species in the group. But obviously, this will have to take a little bit more research to make sure that this is true.
Okay, cool. Hopefully you answered it. Unless if there's a follow-up questions. So the other one is from Albert to Nawa. The question is, considering the damage that the crayfish are causing and the serious impact on livelihoods, do you think it will be feasible to lobby for their eradication from the invaded systems? And another question is, what could be the alternative measure to reduce their impact within the system where they already occur? Nawa? Okay, still waiting for Nawa. Yes, we can hear you now.
All right, thank you. Thank you, Albert, for that question. I think there are two questions that I've seen. The one involving eradication. I think it would be really near impossible to eradicate the crayfish, especially the places which have been invaded, simply because of the rate at which they are reproducing. I think they are becoming quite abundant and spreading very fast. But I think one of the things that could be done to probably manage the population is to perhaps introduce the crayfish as an alternative source of food, maybe to perhaps encourage local people to look at it as probably a source of protein. I know that is going to be a bit challenging because of the perceptions that the locals have around the crayfish. They look at it, I think, from my interaction with a few of the fishers, they really wouldn't like to eat it. But I think it reminds me of a very interesting story of a local person who apparently has been eating the crayfish, and what he told me was that he does own the bar, and at one point he tried to cook them and fed them to the people who came to actually drink beer from his place, and they only learned about the crayfish after they had eaten, and they were saying they were actually quite tasty. So I think it's an indication of the possibility that people had actually learned to eat this crayfish with time. The other thing that could probably be done to help manage those population also is to look at it as a source of food for animals like pigs, because apparently there are, I think, a few fishers and the residents on the plains who have resorted to harvesting the crayfish and giving them to their pigs as a source of food. So I think the main focus should also be looking at ensuring that we don't, in terms of policy and management, ensure that the crayfish does not spread to other places, and only manage them from the places that have been introduced. I think that is how maybe we could move forward. I hope I've answered your question.
Okay. Okay. Now, maybe on a follow-up question from me is, what are there any other means in which the crayfish spread, like spread through human interventions, and are there any plans of modeling where the species are likely to spread in the system or in Africa in general? Because I think they are the studies that are coming up now from like from Taku, and you guys are showing like they are really causing the damage on the fisheries and they are spreading. So that's my question.
Thanks for that question. Yes, I think they are actually, this is one of the areas that we could probably look into in terms of research. There are human-aided method of invasion. I think what usually happens on the floodplain is that fishers go to get their nets. They don't really check them to see whether there are any live crayfish. And so they just, bundle them up in their canoes and then they go to the next fishing site. So through the lack of those biosecurity measures, and I think that's one of the management perhaps the solutions we could be introduced is to come up with these strong biosecurity measures to ensure that as the fishers are fishing and moving from one point to another, their gear is properly checked to make sure that these crayfish are not introduced into new places.
Okay, thank you. Guys, let us not forget to vote, especially the new guys coming from the other session. I think the session is done. We can hand over to Yonela. Thank you.
Welcome to the last session, everyone. We thank you all for attending our symposium and also for supporting our students throughout the day. This symposium was in collaboration with SciFest Africa, and here is the message represented by Mr. Freddie Mashate, the manager of SciFest Africa.
Apologies, and I was just trying to unmute myself there. Thank you very much for giving us the opportunity, and let me just start by saying congratulations for all the great work that you guys are doing. I was listening with great interest, and I think one that just caught me was, when Peter started to speak, because his research is in an area that I live very closely to in the Harib Dam and Ovestone, where we go to fish. So it was really interesting to learn that things that we do as a hobby, could be a point of research and have greater impact. So really congratulations to all of you. I'm just here to first of all thank SAIAB for allowing us to collaborate, to assist in the running of this event, but also to introduce our platform and what we do and the opportunities that are there for you as students and as researchers. First of all, my name is Freddie Mashate, as has been mentioned. I am the manager of SciFest Africa, South Africa's National Science Festival. It is a project that was started in 1996, so we are celebrating 25 years this year, with a specific purpose of creating an awareness, appreciation, and understanding of science, innovation, and technology. And maybe just to simplify this, what we are really doing is to just bridge the gap between science and the public. And I think that is where it becomes interesting and where you guys as researchers come in. Having done a lot of research, great research that you are doing, I think that it would be a lost cause if the public is not engaged, is not informed of the great work that you're doing. And we then provide that bridge. How we do that is through two main ways, or through two main streams. We have, I'll start with what we usually refer to as a second point, but it's outreach programs that we do throughout the rest of the year, including your ESCOM Expo for Young Scientists, if you've heard of them. We also participate in the National Science Week, which happens annually around August. But the other stream is, which I want to just speak a little bit more about, is the annually held festival. Basically, we have a festival that we hold annually. When things are normal, it happens in Grahamstown in Makanda at the monument, 1820 Settlers Monument. But because of COVID-19, we have had to come online. So that offers an opportunity for researchers to come and present the work that they're doing, to interact with the public, to present so that the public is informed, so that they also get feedback from the public with regards to the work that they are doing. So that then is an interesting opportunities. Some of the activities that we do, and especially I'll just refer to some of the activities that we do in this online era, include webinars, which would be talks, panel discussions, presentations such as these that you are doing. We also have workshops, we run competitions. So I'm just giving you this so that you kind of have insight in some of the programming that we have. We have virtual tours. So depending on your work and what you do and what where your research is, targeted at, there is numerous ways that you can participate. Please do visit our website. Just type in Southwest Africa on Google. Our website will come up. Visit our website where you can find some of our past events and some of our upcoming events, interesting events that are already happening. But also very important, there's a form there that you can fill in if you want to participate in the festival. It's called an expression of interest form, where you can just basically fill it in, and we will be able to review whatever it is that you want to contribute, and hopefully we can welcome some of you to the festival platforms as both presenters and also an audience. So a lot of exciting stuff. So I was really not here for long, but just to hopefully introduce you to this platform that might be of interest. And especially I think when we speak about engaged research, many of you would know that a lot of your funding now requires you to have that aspect where the public is involved. And what we have also done is to work with Rhodes University, and in particular the environmental science department, where we try and find ways to engage the public in your research and ensure that there is that bringing together and bridging that gap between science and the general public. So yeah, our doors are open. Please do come through and speak to us, and hopefully we can work together going forward. Thank you. Thank you very much, Program Chair.
Thank you, Mr. Mashate, for that information. Hopefully our students will sign up for this. We're now moving forward to the student prizes. We can all agree that all presentations were good, and to us, you are all winners. But just for today and for formalities, we'll only present the best prizes for the MSc and the PhD. The winners will walk away with the book, The Fishes of Zimbabwe and Their Biology by Brian Marshall, and also a signed copy of the recently published Fishes of Southern Africa, Southern African Estuaries from Species to Systems by Alan Whitfield. We would like to thank the Margaret Smith Library, Professor Whitfield and Ms. Melthouse for sponsoring our prizes. We're now crossing live to the second floor. Luba is ready to announce the winners.
Hi guys, I'm just waiting for the final outcomes. They're busy doing final calculations. So, okay. So the winner for master students is Craig Rennie, and the winner for PhD is Yonela Sitole. So yeah, congratulations guys. So that's it from me, and then I'll give Albert Chakona for some closing remarks. Thank you, and well done again to the winners and to all the presenters. Thanks a lot, everyone. Cheers.
Yeah, thank you, everyone. My job is very simple, is to just thank everyone. So I just want to first thank our guest speakers, Monica, Jenny, Elisabeth, and Melanie, for their fantastic presentations. Thank you so much for sharing all that information with us. And then I want to thank all the presenters, the team leaders, the session chairs, the Margaret Smith Library already mentioned, for sponsoring and organizing the prizes. And then all of you for attending. And then finally to the team that made all this possible. So the postgraduate students and postdocs in the ecology and ecology, evolution and systematics group. Well done, team. We're really proud of what you've produced. And then the SciFest Africa team for hosting and facilitating this whole program, Ryan, Nikki, Jim, Akona, Darren. And then finally, Dr. Angus Patterson, our managing director, for all the support and guidance. Thank you, everyone. I wish you very good holidays and keep safe. Thank you. Thank you, Program Director.
Thank you, everyone. That's it for today. We'll meet again next year around this time.